Well, boys, this is central dogma again. We will be discussing elongation. But before that, let's just do a little recap. Central dogma, it comprises of transcription, translation and protein synthesis. This is the flow of information. From DNA, mRNA is produced, and from mRNA, proteins produced. Okay, so we started with this one, transcription, and transcription consists of initiation, elongation and termination. So this we've done in another video, previous video, okay? So today we shall be dwelling with elongation. And what we saw in initiation was that the DNA inside the nucleus is fixed with a polymerase, RNA polymerase. Okay, and this is RNA polymerase 2, which produces mRNA. RNA polymerase 1 produces rRNA, and RNA polymerase 3 produces your tRNA. Okay, so these are things that we've done in the previous videos. The process of elongation is the formation of the long peptide chain with the addition of nucleotides. Okay, this results in the formation of pre-mRNA. Pre-mRNA meaning precursor mRNA. So this is what happens in initiation. The latching up of the RNA polymerase. I'll just write RNA pol 2. Okay, and it latches on to the promoter, which is present on the DNA. Okay, this promoter is also known as the Tata box. So just beside it, it links to the promoter, and then begins to move forward. As it moves forward, it splits the DNA into two. Okay, and the strands are separated this way. And as it is separated, it begins to form this long chain of mRNA. So it begins to copy one of the strands. Now this copying of the DNA is a little tricky to understand. In order to understand that, we have to go through this again. This is the pentose sugar, and we know that it has 5 carbons, and these are the carbons here. Okay, and it's 1, 2, 3, 4, and 5. Important to remember, on the 5' end of the carbon, we find the phosphate group. And to its 3' end, it has the OH, or the hydroxyl group. Okay? So, the addition can be done this way. You can, the pentose sugar, this is the pentose sugar, this is the nitrogenous base, and this is the phosphate group. Phosphate group, pentose sugar, nitrogenous base. So this will be fixed on this way, okay, here. So once it's done, once this is added, you get a structure like this. Another one, okay, and so on. So this makes the one single strand of the DNA. So the point is this. Once you add another nucleotide, this becomes your 5' end, and this the 3' end. Okay, so you can keep on adding nucleotides to the 3' end. Okay, so with that as background, we come here. This was the original DNA here. Okay, once it's split apart, so now we have the 2 strands here. This is the upper one, this is the lower one. Okay, now these were held by hydrogen bonds, and it runs anti-parallel. Okay, 5' here, 3' this side. On the other strand, it's the opposite. 5' this side, 3' on the other side. Okay, and it faces opposite direction. So that's known as your anti-parallel property of your DNA. Now what happens is, once it's broken apart, or once the DNA strands are separated, and the Y fork for copying of the DNA is produced, the same polymerase begins to copy. Now, if you check this out, if this strand is taken, okay, and then if you want to add, since this is being broken up this way, mRNA is being copied from left to right. mRNA copied, or let's say made from left to right. In this case, okay, left to right, because this is being opened up this way. This side is being opened, this side is still closed. So it opens up, continues to open up, and then it also adds up new nucleotides to one strand, whichever strand is running from 3' end to 5' end. It cannot add to the 5' end because this is being opened this side, okay? And as it goes this side, it cannot add another one this way. It's easier for it to add in. So here's one nucleotide which can be added this side. Okay, it cannot be added onto the 5' end. It can be added only to the 3' end. So it goes this side, but this is being opened on the opposite direction. And if you keep on adding nucleotides this side, you end up hitting the promoter. Okay, and that's the end. So it's copying this much and leaving behind all of these, the newly opened up codes. So this is not copied at all. So this is called the non-template strand. Whereas on the other one, the bottom one, which runs from 3' end to 5' end. This is the 3 and this is the 5' end. Okay? So here it's easier to add. You can just bring it here on its 3' end. Another one on its 3' end again. So as this is being opened this side, as it progresses this side, you can continue to add nucleotides like that. So this can be, this end, this template, or this strand can be copied more easily. So this is called the coding template. Or you can say the template strand. You can just call it template strand. So this is the one which is copied. Okay, as it progresses furthermore towards the right hand side, as it continues to open up, you can keep on adding the nucleotides. The other way around. You can keep on adding these nucleotide pieces to make this mRNA. So this is the pre-mRNA. Okay, this is the promoter, and this is your RNA polymerase 2, which has two works, not just to open up. One is to open up or split up the DNA, and the other is to add on nucleotides to form what is known as the pre-mRNA. So this is what elongation is. The important thing to remember is addition of nucleotides can occur only in one direction. Therefore, it's easier to copy. One of the strands is easier to copy as it opens up on one side. The other, it has to be copied with, small pieces needs to be copied. First this one, then it opens up till here, and another one needs to be copied. So only segments of it can be copied on the other side. So that strand is now called non-template strand. Whereas the one that can be copied easily as it opens towards one side from left to right, that strand is called the template strand. Okay? So the elongation process is the addition of nucleotides to form pre-mRNA. Right?